Hello, nice to meet you. Today I'll be presenting my work from my PhD, actually. The title of the talk is called Germline and Somatic Mutational Processes Across the Tree of Life. The project was supervised by Peter Campbell and Richard Durbin, and was leveraging the public data sets available through the Darwin Tree of Life. Just want to give a brief overview of the Darwin Tree of Life project. The Darwin Tree of Life project is based at the Wellcome Sanger Institute and aims to sequence every eukaryotic species in Britain and Ireland. Currently, the program lead is Mark Blackster. The experimental design is quite simple. We collect a couple of species, but we sequence one sample per species. And we make the data set and the reference genome almost immediately as possible when they're ready. And we are currently using hi-fi reads primarily for de novo assembly. And we are using Hi-C scaffolding for scaffolding of the contigs and for the manual curation of the scaffolds into chromosomes. And if you're interested, all the data sets and the reference genomes the status of them are available through this website. And before we go further, I need to make some definitions and kind of things that may be unfamiliar with people that are not in the cancer genomic literature. First, everyone here will know the differentiation between germline and somatic mutations. But for the sake of argument, germline mutations, anything that happens in the gametes and that is inherited by all the cells of the organism. And somatic mutations happens in the soma and is limited to the pocket of a tissue. And in this study, we attempted to sequence both the mutational processes that impacts the germline and the somatic tissue. And before we go into the meat of things, we also need to understand how typically substitutions are classified in cancer genomics literature. On the left-hand side, we have the substitutions in the purine context, and in the right-hand side, we have the substitutions in the pyrimidine context. And in cancer genomics literature, we typically use the substitutions in the pyrimidine context, and then to add some additional classifications, we add the trinucleotide context to each substitution to further classify it. And if you have read any of the cancer genomics literature, you might have seen a mutational spectrum plot like this, and typically we have the trinucleotide context on the x-axis, and we have on the y-axis the proportion of mutations this particular mutational process might produce. And here we have an example of a mutational process we call as SBS1, and that typically is a representation of the spontaneous deamination of methylcytosine to thymine at NCG trinucleotides. And the peaks there are all from the deamination of methylcytosine to thymine. And my supervisor, Peter Campbell, has been interested in looking at normal tissues and tumors to understand the mutational processes in human tissues that generates driver mutations and where those driver mutations might lead to tumorigenesis. And that study has led to the discovery of about 49, 50 somatic mutational signatures, each representing a distinct mutational process. And because of the availability of the Darwin Tree of Life, my supervisor, Peter Campbell, started asking, so then what's the mutational process that is governing the, in non-human samples, basically? And we therefore then needed an attack vector to attack that question of answering what is the mutational processes across the other samples. And I'm sure many of us here know that CCS reads are very, very accurate. But to illustrate my point, so typically we have something called duplex sequencing that uses unique molecular identifiers to cluster sequences and generate a consensus sequence, and that is typically used in somatic mutation detection. And CCS sequencing in PacBio is very, very similar. It doesn't have a unique molecular identifier, but a single molecule is sequenced within a ZMW, and the redundancies of sequencing and the complementary base pairing between the double-stranded DNA is used to generate a consensus sequence. So the natural hypothesis was then, duplex sequencing used for somatic mutation detection, can we then also use circular consensus sequencing for somatic mutation detection as well? And there's really three scenarios where this can happen. So the first scenario is where sequencing error rate is higher than somatic mutation rate. The second scenario is where sequencing error rate is similar to somatic mutation rate, And the third scenario is where the sequencing error rate is lower than the somatic mutation rate. And in the first scenario, it's not going to work. This project is not going to work. In the second scenario, if the sequencing error rate is similar to the somatic error rate, mutation rate, then you can potentially profile the mutational processes that are actually happening in the cell. And in the third scenario, the sequencing error rate is so low that you can start detecting somatic mutations from a single observation. So to tackle this hypothesis, we developed a method called HiMIT. It is available as an open source software. So if you're interested in using this software after hearing about the research, do check them out on my GitHub page. But basically what it does is it tries to distinguish sequencing errors from somatic mutations using some of the features that we have identified. And it requires unbinned CCS reads with unbinned-based quality scores to detect somatic mutation. And we go through a process of distinguishing germline and somatic, and then distinguishing sequencing errors from somatic mutations. And at the end, we find a somatic mutation that here is highlighted in green circle, where a single observation is sufficient to start profiling the mutational processes across the samples across the tree of life. And kind of to demonstrate this point, we first sequenced some bulk normal tissue from human cell lines and cancer cell lines to demonstrate our point. So first, we sequenced 2 cancer cell lines. One is called BC1, one is called HT115. And the reason why we sequenced these samples was because we knew the mutational processes that are active in these samples and the mutational spectrum that is produced by the mutational process. In the BC1, ApoBec mutagenesis generates the cytosine to thymine substitutions, and the HT115 sample, poly mutagenesis, is responsible for the mutational spectra that you're observing. And then we also sequenced some blood, blood from a 77-year-old and blood from a fetal blood, cord blood. The reason we sequenced the cord blood was because the number of somatic mutations that is acquired in your sample is dependent on age. And if you sequence a very, very young sample, obviously the number of somatic mutations that would have been acquired is very little. So it acts as a negative control in our experiment. And then we started applying our method. So we got some BC1 cell line, we got some HT115 cell line, and sequenced them, applied our method. And as you can see, if you can see the left and right, the mutational spectrum that is observed is highly similar, indicating that we are able to actually observe the mutational spectrum produced by the mutational process specific to the sample simply by doing some somatic mutation calling from those samples. And similarly, we obtained some blood from an 8-year-old and another independent cord blood, and we can similarly see again that the mutational spectra that is observed in the 77-year-old and the 82-year-old is very, very similar, indicating that this is also possible in bulk normal tissue. But the error rate isn't sufficient to detect somatic mutations accurately in samples where the acquired somatic mutation is a bit too low, as you can see from the comparison of the 2 cord blood mutational spectra. So knowing that, we continued. And this is the basic experimental design of the project. The Darwin Tree of Life consortium started collecting some of these samples across Britain and Ireland, and I did not have to do the hard work, and I'm really thankful for that. And we sequenced a bunch of eukaryotic species, insects, plants, animals, anything that we can get our hands on through collaborations. And we subjected them to circular consensus sequencing, and then started applying our method, HiMIT and deep variant, to cold germline and somatic mutations. So the natural question would be, yes, you have demonstrated that this works in humans, but can you demonstrate that this works actually in non-human samples as well? So to demonstrate that, typically we sequence 1 sample per species, but in this case, we started to sequence multiple samples from each species to identify or to show that the mutational spectra that you're observing in species-specific mutational spectra is independently observed across biological replicates. And here we have a 2-spot ladybird, very, very adorable, and then we had multiple biological replicates, but here we show 3 biological replicates where the same mutational spectra is observed. And then if you do a UMAP clustering of these mutational spectra across samples where multiple samples were a species across, where multiple samples were sequenced, we can see that these samples clustered very, very nicely away from each other depending on the mutational spectra that they have. So we're starting to gain more confidence that, okay, this method works, and we're starting to observe or kind of directly observe the somatic mutational processes across the tree of life. So we started looking at 570 eukaryotic species across 600 samples, across different taxonomic classifications. We sequenced fish, mammals, mollusks, Lepidoptera, Diptera, and so we started using every sample that we could get hands-on. And we identified 22 germline mutational signatures, and we identified 98 somatic mutational signatures. I will not go into every detail of every signature, but we are also hoping to submit a manuscript of this paper very, very soon, so look out for that. But here is just one example of a mutational process that we identified in an insect called the thick-legged hoverfly. And in this particular example, we have mutations that are specific to a cytosine to adenine transition in the sequence context of NCG. And if you actually perform a phylogenetic analysis of these mutational signatures, we find that the mutational signatures are clustering based on either their environment, taxonomic classification, or the ancestor that they share. So here we have a phylogenetic tree based on the 600 samples that we have. At the middle, we have the common ancestor that is shared by all the samples that we have. And we have colored the species that we have by their taxonomic classification. In green, we have the plants, and in the other colors, we have the insects. And this particular signature that we have called STOL6, and we have in green the kind of the phylogenetic signal that we have measured for this particular signature, and you can see that this particular signature is overly represented in plants, for example. And in this particular signature, STOL14, this signature is more strongly represented in the insects. So this is just one example of mutational processes that are shared across different taxonomic classification. So some of the, I guess, take-home messages that I wanted to share is that I think still this is kind of an idea that still needs a bit of convincing for the field, but the core message that I wanted to deliver was that if you really kind of start subsetting the hi-fi bases that you have, you can actually start using those hi-fi bases to profile somatic mutational processes across the tree of life, from bulk normal tissue, agnostic of species and clonality. So if you have like any genomes that you're interested in, and if you want to actually study the somatic mutational process of that sample, just apply the method and you'll be able to see what's the somatic mutational process that is in that sample. And there are some caveats there with life cycle of each species, but maybe that's a separate discussion. And in humans, we had to study normal tissue tumors to identify 49 signatures, but from sequencing hundreds of samples across the tree of life, we find a rich array of mutational processes that we have never discovered before. And I think this is going to be a fascinating area of research in the future too. And there are some discussion points that I guess will be relevant to continued future research, but we've discovered many new mutational processes, but we have not even started scratching the mechanism that is generating these mutational processes. So that will be another fascinating area of research. And maybe this is not relevant specifically to the audience, but if you apply this particular method of experimental design to population scale sequencing, I think you'll be able to find individuals that are impacted by environmental mutagenesis as a result of involuntary consumption of some sort of mutagen or carcinogen, and start identifying individuals with DNA damage repair deficiencies. And we also have another exciting paper that's going to be coming up in a month that is also using CCS sequencing, and this knowledge of the fact that hi-fi bases are really, really accurate allowed us also to investigate meiotic recombinations from bulk sperm sequencing. So that's another paper that's going to come out soon. So do look out for that. And just wanted to say, send some acknowledgements to my supervisors and colleagues and funding bodies. Peter Campbell initially conceived the idea, but both Peter and Richard Durbin kind of supervised this project, and they have been unbelievable. And Wellcome Sanger Institute funded the project. I funded my research PhD, and University of Cambridge and Darwin Tree of Life. And the Wellcome Trust has been an unbelievable help for the PhD. Well, actually, not question, necessary. Thank you very much.
Thank you, Sangjin.